We are Senior Design Team 10, and our project is radiofrequency ablation modeling and validation in non-homogeneous structures. Radiofrequency ablation is a minimally invasive procedure that's meant to alleviate pain. The process involves an electrical current that heats and burns small sections of nerves in order to interrupt the pain signals sent to the brain. During this project, our sponsors at Medtronic provided us with their Colaianne RF ablation system for testing. The following picture is of the Medtronic Colaianne system, showing the console, the wires, and the probes used with the system. Predictable lesion formation in non-homogeneous tissues is of particular interest in radiofrequency ablation, so our team was tasked with developing mathematical models and physical model validation for such treatment scenarios.
In order to create a digital model that can accurately predict the lesion size and temperature distribution of the radiofrequency ablation procedure, Team 10 was able to use COMSOL multiphysics software. To get a better understanding of this procedure within non-homogeneous tissue, a control model of sorts was built. Within this control model, there are two test cases. The first is the muscle-only case present in the top half portion of the model. The second is a bone cylinder case, which is present in the bottom half portion of the model. The temperature distribution and estimated lesion size for each case are presented. As can be seen, the length of the lesion was not as affected as was the width in the lesion. Moving on from the control model, Team 10 was then able to create a more accurate representation of the human knee and what the procedure would look like. Pictured below are the human knee model results in the form of an estimated lesion size, as well as a temperature distribution contour. Physical validation testing will be an efficient way to determine whether or not this human knee model is accurate.
A physical model is created in order to verify the digital model. For model validation, lesions needed to be created and measured. A measurement system was developed where tissue would be secured in a fixation box during ablation, tissue slicing, and measurement. Multiple operators would take several measurements. This system was verified using a reproducibility test that was designed based off ISO standard 21748-2017. The figure on the left shows the ablation setup. On the right, it can be seen that there is a formed lesion. The lesion is shown as a cooked portion of the tissue, so it is light in color. In order to determine the effect of bone presence on lesion size, tissue with bone was ablated with several different probe alignments and electrode placements. Lesions were measured in every instance. The figures below show three simplistic models of different trials that were done. The results of this study show that lesion length decreases by 9 to 28%. Lesion width remained the same in all trials. The conclusions reached from the physical model are that the presence of non-homogeneous structures reduces lesion length.
Due to the outbreak of the COVID-19 virus, much of the testing for the project was cut short. It is recommended by the team that if this study is to continue, more trials are performed to better assess the effect of both probe alignment and electrode placement on the size of the lesion. It is also recommended to explore bipolar ablation to determine if lesions can be created with better predictability than with the monopolar ablation.